When we're talking about epistasis, we're not talking about just one protein that determines the phenotype. We're talking about the interaction of two or more sort of different genes, so different chromosomes, that have an effect, a unique effect on the phenotype, and they sort of interact with each other. So, I mean, the most common examples are in colour, because traits, I mean, most sort of traits are more complex than just two proteins involved. You've got thousands, you know, absolutely loads of different proteins and biosynthetic, like biopathways in your cells, that there's so much complexity that you can't sort of say, oh, this gene does this and this gene does this, and, you know, and then if we change this then this will happen. It's a giant sort of web of interconnecting things, and it's all very, like, complex. But with epistasis, it's one of these sort of more multiple gene interactions that we can sort of get our head around a little bit, hopefully. By the end of this video you should. So if we are talking again about flower, if we're talking about two genes, so I'm going to say that this enzyme A turns this colourless precursor molecule into a red pigment, and then enzyme B, which is coded for by the gene B, turns that red molecule into a purple one. So we're getting a colourless molecule, and if we have the right enzyme here, that converts it to a red molecule, and if we have the right enzyme here, that'll convert it to a purple one. Now if we have a non-functional enzyme here, this can't be converted to red, and so whatever we have at this one, whatever sort of allele we have here, doesn't make a difference, because we can't ever get to the stage where this becomes important. So this gene is epistatic to that one, because whatever sort of allele is at this locus determines whether this one will be expressed or not. So if this one happens, if this enzyme that we have doesn't turn our white molecule into our red one, then even if we had a enzyme that could turn the red one into a purple one, it won't turn it into a purple one because it wasn't there to begin with. So if, when it's dominant, A codes for red, dominant B codes for purple. So wherever we have a big red and no big B's, then these are going to be red. Wherever we have a big red and a big B, we're going to get purple flowers. And then wherever we have little a, they're going to be colourless, because if we look back, if we have, we're saying that little a codes for a non-functional enzyme here. If that can't be converted to red, then no purple can be made. So even though these three sort of genotypes have the correct allele to give a purple colour, they can't, because there's no red molecule to convert to purple. 9:3:4 ratio. This is recessive epistasis, because when it's recessive, little a, we're not getting the opportunity to express the second locus. So this is recessive epistasis, where little a blocks the effect of whatever's at gene B. So if we were looking at dominant epistasis, where little a codes for red, then wherever there's little a, we can get the possibility of having red flowers. And then wherever big B is, they'll be purple. So if big A doesn't work and it doesn't let us go to the next stage, then all the ones with big A are going to be white, because we can't ever get to the point where it codes for it being red, purple. So we end up with the dominant epistasis ratio of 12:3:1. The other one that you might come across is when we call it complementary. So here we've got the same sort of situation, but except instead of we are going from a colourless pigment molecule to a coloured one and then a different coloured one, we've got two colourless pigments, and then gene B codes for this one to turn into red, but it can't turn this one into red. Only if this locus is dominant and this locus is dominant, then we can get the colour, otherwise we end up with colourless flowers. So when it's complementary, so you have to have a dominant one of both, and those ones will be red. And this is when we end up with our weird 9:7 ratio. So, I mean, it's very, very clever epistasis. You can look at it in lots of different contexts. So this way I've said about different about sort of coding for enzymes in a specific pathway. That is one epistatic mechanism, sort of, that can give rise to different phenotypes by gene loci sort of interacting. The other way, well, one of the other ways that gene loci can interact is sort of in terms of repressor proteins. So if we have two genes, gene A and B, and gene A codes for a protein which stops gene B being transcribed, then whenever we get the functional version of that protein, we won't get any gene B. Whereas if we get the recessive version and it can't bind properly, then we can get the effects of gene B. So that's another way of it operating. It could be that one protein is responsible for degrading another one, or is a cofactor to something else, and then that's another gene sort of, gene product interaction is the products of the genes that interact as opposed to the actual genes. But we always talk about it genes because when we look at it in inheritance, we're talking about the gene. Anyway. So that's another way it can interact. It could be that one protein activates another one. So if you have a protein B, but it has to have another protein bind to it in order to be able to catalyse the reaction. If this one is non-functional and can't bind, whatever we've got at B isn't going to work. If it is functional, then yeah, B will work. But maybe because it's got two different alleles, one will have a nice sort of active site which will turn it to one thing, and the other one will have one which turns it to something else. So there's loads and loads and loads of sort of variation in the mechanisms of epistasis, but most of sort of the genetic variation that we get is from far more complex mechanisms. So you can see just with two genes, the way that they interact, the way that proteins interact in cells, the way that sort of you have so many different factors that bind to DNA and affect the transcription, or even the translation, binding to sort of proteins as they're being made and sort of affecting that. You can see there's so much room for loads of alterations and regulation to occur. And I'm going to do a video on that because it is the coolest stuff ever, and I haven't done it yet. I'm very sorry. But yeah, that's epistasis. If you have any questions, ask them. Okay, bye.